My name is Leen. I'm the Chief Scientific Officer, and today I'll be telling you about AlloVir, a company dedicated to advancing off-the-shelf virus-specific T cells for the treatment of devastating viral infections. Here is our disclaimer. And moving on to really introduce you to who we are and what we're doing. We are a leading innovator in the use of allogeneic off-the-shelf virus-specific T cells with significant clinical trial experience in patients with infections. Indeed, to date, we've treated well over 275 patients with our therapeutics and achieved extremely promising data, including most recently a phase 2 clinical trial where we achieved a 93% response rate with our most advanced product, viral when it was administered to transplant recipients with refractory viral infections. And indeed, the same product has received both RMAT and PRIME designation from the FDA and the EMA, respectively. Our pipeline is extremely rich, and as the Chief Scientific Officer, it's my job to really build out that portfolio of products. And to date, we have a program with up to 9 pivotal and proof of concept trials planned with our different therapeutics and the potential to access an extremely large global market. What really supports our company is our deep expertise in manufacturing these off-the-shelf virus-specific T cells, which can be generated at extremely large scale to really support the global market that we serve, that we intend to serve. In addition, our company is really supported by our robust IP and patent portfolio, which covers both products and our processes for generating and testing our virus-specific T cells. As I'll introduce to you in a little while, we have an extremely strong investor base. And as I previously detailed, we have a very experienced and proven team, both from the business side, clinical side, manufacturing, and commercialization. On the next slide, to speak to the team specifically, we're led by David Halal, our Chief Scientific Officer, and Vikas Sinha, President and Chief Financial Officer. As I mentioned, I'm Chief Scientific Officer alongside Tino Melian and a team with deep regulatory and business expertise and extremely deep expertise in manufacturing. We're really advancing these therapies to the clinic as fast as possible. We are supported by a very strong group of investors, some of whom are detailed at the bottom of this slide. To speak to the issues that we're trying to deal with our therapeutics, we're really trying to cater to high-risk patient populations who are deficient in their immune system, and specifically their T-cell immunity, which really renders them vulnerable to a whole spectrum of life-threatening viral diseases that are persisting and problematic despite other therapeutic options, including conventional therapies. The viruses that we're seeking to target with our off-the-shelf therapeutics are shown on the left-hand side of the slide and include both latent viruses such as BK virus, cytomegalovirus, EBV, and human herpes virus 6, as well as a variety of lytic viruses and respiratory viruses such as RSV, parainfluenza, human metapneumovirus, influenza virus, and of course the virus that we hear a lot about these days, SARS-CoV-2. These different viruses impact a variety of organs in the body, and that's really showcased in the center of this slide, where we see impacts in the bladder. For example, BK virus can result in severe hemorrhagic cystitis, urinary obstruction, and even can result in patients requiring cystectomies. But equally, a variety of different organs and other organs are impacted by these different viral infections and diseases, including kidneys, lungs, intestine, brain, eyes, liver. And of course, we're even treating some viruses that are associated with malignancies. And on the right-hand side, the different patient populations that we're seeking to serve. These are a spectrum of individuals, including those who are compromised due to treatments that they receive, including bone marrow transplants. And so those are the patient population where I previously noted we'd achieved a 93% response rate. But equally, we're seeking to serve other patient populations, including recipients of solid organ transplants and other high-risk and vulnerable patient populations, including the elderly, very young, and those who are receiving treatments for cancer, for example. Here on this slide, I'm showing the pipeline that we have of products that we're advancing to the clinic. As mentioned, this is an extremely deep pipeline. Indeed, we have 5 allogeneic off-the-shelf therapies targeting a total of 12 viruses overall. Noted previously, our most advanced product is viral MM, which we are rapidly advancing to a number of pivotal and registrational studies for a variety of indications, ranging from virus-associated hemorrhagic cystitis to the treatment of CMV and solid organ transplant recipients. We also have a second multi-virus targeted T-cell therapeutic directed against a range of respiratory viruses such as RSV, flu, paraflu, and human metapneumovirus, which will be moving to the clinic for the treatment of infections and diseases associated with these viruses, both in the stem cell transplant setting and the high-risk general population. Furthermore, we have a number of other products where we have generated therapeutics, for example, to treat COVID-19, again in high-risk patients. That's our ALVR 109 program, with more early-stage products targeting HPV and HHV8. But for the purposes of the remainder of this presentation, I want to really talk to you about our most advanced product, which is viral MM, which we designed to treat 5 viruses that are extremely problematic, particularly in the stem cell transplant setting, and to really showcase the magnitude of the response, the magnitude of I think the need for a therapeutic in this space. What this slide shows actually is data generated from the Fred Hutch in Seattle, where they looked at the incidence of infections associated with the viruses that are targeted by our viral MM product, namely BK virus, CMV, adenovirus, EBV, and HHV6. And if you simply look at the incidence of these different infections in the first 100 days post-transplant in the allogeneic stem cell transplant population, you'll see that almost 90% of all patients have issues with at least one of these target viruses, and almost 2/3 have issues with 2 or more of these viruses, really speaking to the profound need for a therapeutic in this space. And notably, there's a 37% increase in non-relapse mortality for every log increase in viral load from day 1 to 100 in allogeneic stem cell transplant patients. So again, showcasing the magnitude of the importance of these infections in this patient population. So one thing is the incidence of infection, but what does this really mean to the patients when they develop these infections? And it's really the fact that these infections can progress from the infection state to causing diseases in these patients very rapidly. For example, hemorrhagic cystitis, a very common manifestation in the stem cell transplant setting, can be caused by viruses, including BK virus, adenovirus, and to a lesser extent CMV. And when a patient experiences or develops hemorrhagic cystitis post-transplant, how they experience that really is with the severe, prolonged, and intractable pain associated with the infection. When the disease occurs, it can cause severe bleeding due to hematuria. These patients experience life-disturbing urinary symptoms that can ultimately result in kidney dysfunction and even failure over time, all culminating in an increased risk of mortality in this patient population. And conventional therapy for hemorrhagic cystitis post-transplant is simply supportive care measures. So these patients receive narcotics to try to control the pain. They have to receive transfusions to deal with the severe bleeding and try to really redress the balance there. Ultimately, they can require bladder embolization or indeed cystectomy if the disease is prolonged and the patient continues to have uncontrolled problems. Ultimately, oftentimes these patients require continuous bladder irrigation, and in some cases can require dialysis depending on the level of kidney dysfunction that's seen. But these problems are just not limited to BK virus. For example, CMV, which can affect up to 65% of all allogeneic stem cell transplant recipients, can also result in potentially life-threatening consequences when advancing from the infection to the disease state. And the diseases associated with uncontrolled CMV infections include pneumonia, colitis, retinitis, encephalitis, and even in cases can lead to multi-organ failure and death in patients. Equally, adenovirus, which disproportionately impacts the pediatric population, can also result in life-threatening consequences ranging from pneumonia, hemorrhagic enteritis or cystitis, hepatitis, multi-organ failure, and again, oftentimes leads to death. And for neither of these viral diseases are there FDA or EMA approved antiviral agents. Of course, on the clinical side, the clinical teams don't do nothing when these patients experience these infections that result in these life-threatening consequences. And so they do use off-label antivirals. The problem there is, though, that the efficacy is often quite limited, even questionable, and the toxicities associated with these conventional antivirals can be severe, including resulting in nephrotoxicity, myelosuppression in cases. And as some of the audience may know, fairly recently there was a prophylactic agent approved for the prophylaxis of CMV, but ultimately this is approved for the first 100 days post-transplant. And recent studies have shown that once letermovir was stopped in these patients, the actual CMV issues recurred. And so really what it does is push the problem down the road, but it doesn't actually redress the underlying issue which is present in these patients and really renders them susceptible to these life-threatening consequences. And that is the fact that these patients don't have an effective immune system of their own to deal with these viruses. Lest you think that infections are solely a problem in the stem cell transplant setting, here is some published data just looking at the impact of these infections in the solid organ transplant situation. On the left-hand side, we're showing some data here for patients who are recipients of kidney transplants and develop a BK virus-associated nephropathy post-transplant. And as you can see in the red line, patients who develop BK nephropathy have an increased risk of graft loss associated with that particular virus. And on the right-hand side, a similar picture, but this time showing the consequences of persistent CMV infections in the solid organ transplant space, again, which leads to an increased risk of graft loss and ultimately an increased risk of mortality. And so all this to say, these infections are frequent, they are associated with significant morbidity or mortality and are not served by conventional antivirals, all of which led us to explore whether we could use virus-specific T cells, which were prospectively generated, cryopreserved, and utilized as an off-the-shelf therapeutic to deal with these different infections. And the reason to think that this is the right approach to deal with these infections is showcased on this pictorial slide here. Ultimately, the patients that are at highest risk for devastating viral infections are patients whose own immune system is somehow deficient or defective and not able to deal with these viruses on their own. And that really renders them susceptible to life-threatening consequences. And so therefore, why not go ahead and build, if you like, immune systems for these patients and have them prospectively prepared, readily available as an off-the-shelf therapeutic to deliver to these patients to really redress the balance and to enable these patients to fight these infections on their own. And that's showcased on the right-hand side of this slide here. We have done this now, and as I previously showed you, we have really quite a deep pipeline of products in both clinical trial testing and in development stages for treating a spectrum of these different viruses. But the reason we're really so well placed to be able to advance these therapeutics is shown on this slide. And it really highlights how efficient and industrialized our platform is and how deep our experience and expertise is in this space. On the left-hand side, what I'm highlighting is our deep experience in being able to profile clinically problematic viruses to identify which components of these viruses we should target with our off-the-shelf therapeutics. And indeed, we have proprietary elements filed around our ability to be able to profile these viruses and advance the correct elements of these viruses to our manufacturing process to be able to manufacture T cells that are protective in nature. We also have proprietary know-how and expertise filed around our ability to be able to identify the appropriate donors from whom to generate these virus-specific T cells from. Remember, our product is an off-the-shelf therapeutic, which we generate from healthy donors who are allogeneic donors, and who have really the genetic profile and HLA characteristics from whom we can generate these virus-specific T cells to be able to serve our patient population. And again, as a company, our commitment is to be able to serve greater than 95% of all patients who could potentially benefit from our therapy with the products that we generate and release for off-the-shelf use. Our manufacturing process then, as shown in the middle of this slide under step 2, is extremely rapid, very scalable. As I said, we can really manufacture these virus-specific T cells at levels that are required for being able to serve our global patient population. For example, for manufacturing of our viral MM product, we have identified a combination of antigens from each of our target viruses, which we expose to peripheral blood mononuclear cells from our carefully chosen donors, as showcased in step number 1. Our manufacturing process takes approximately 2 weeks without interventions during the time frame between initiation and cryopreservation at the end of our manufacturing process. And again, as I alluded to, these cells at the end of our manufacturing process, which are highly enriched for our virus-specific populations, are harvested and cryopreserved. They maintain their stability long-term and are thus available on demand as needed for patient use. And essentially, each manufacturing run that we perform from a single donor can generate hundreds of doses of these virus-specific T cells. Our ability to be able to rapidly deploy these cells as needed is also showcased by a second software that we developed to rapidly identify the correct donor-derived allogeneic virus-specific T cell product for each individual patient, and that's called CytoMatch. And again, this enables us to be able to be immediately responsive to clinical needs as and when we are approached for patients in need of our therapies. I want to go into a little bit more detail about our viral MM product. This is a multi-virus product that we developed to essentially fully transform the lives of transplant patients who are at risk of multiple viral infections post-transplant. To give you some more nuanced detail about this product, this was a product that we designed to be able to deal with the 5 viruses that are most frequently detected and are most clinically problematic in the immediate post-transplant period when patients are immune deficient. And these 5 viruses are BK virus, CMV, adenovirus, Epstein-Barr virus, and human herpes virus 6. To target each of these viruses effectively, we identified at least 2, if not more, immunogenic viral antigens within each of these viruses against which to raise our virus-specific T cells. And indeed, the viral MM products that we generate are actually reactive against 5 viruses and a total of 12 selected antigens from each of these 5 viruses. We use these immunogenic antigens. We expose our donor peripheral blood mononuclear cells to these antigens, and over our 2-week manufacturing process, we effectively stimulate and selectively expand out these virus-specific populations to yield a multi-virus specific T cell product at the end of that process, where we have subpopulations of cells targeting each of the 5 viruses. To date, we have recently completed a phase 2 study, proof of concept study of viral MM in our population of allogeneic stem cell transplant recipients. This phase 2 clinical study was called CHARMS, and indeed we published the results of the first 38 patients who were administered with this product in 2017 in the Journal of Clinical Oncology. And today I'll really be bringing you up to date in our total clinical trial experience of 59 patients treated. At the beginning of this talk, I really spoke to you about the incidence of these infections post-transplant, but I want to speak in more detail to the fact in this particular clinical trial, we really want to very effectively test the activity of our product in patients who are in dire need of therapeutic intervention. And for that reason, our proof of concept phase 2 study was performed in the allogeneic stem cell transplant patient population, but specifically in patients who had infections with one or more of the target viruses, and in whom these infections had proven to be refractory to conventional antivirals that had been applied to treat these infections, or where the patients who had been shown to be unable to tolerate the off-target toxicities associated with these antivirals. And so suffice to say, these were really quite a sick patient population who were in significant clinical need of therapeutic intervention. In these patients then, we administered our viral MM product. This was a fixed cell dose of 20 million cells per meter squared. And if the patients did anything less than achieve a complete response to therapy, they could receive up to 4 additional cell doses at 2 weekly intervals from week 4 post-infusion. The study endpoints were primarily safety, to assess the safety profile of our product in this patient population. But of course, we looked very closely at evidence of clinical efficacy, also looking at both the viral load in these patients as well as clinical signs and symptoms. To speak first of all to the safety profile of this product in these patient populations, it was extremely well tolerated. Here on this slide, I'm just summarizing all of the safety indicators. We had 3 patients that developed an isolated fever within 24 hours of infusion, but otherwise, all 3 of which resolved without therapeutic intervention, and no other immediate toxicities were observed. We also looked, of course, for any evidence of graft versus host disease associated with our therapy. In summary of our 59 patient experience, we had 6 patients who developed de novo graft versus host disease, but in each case, this was the mildest grade 1. It was all of the skin and all resolved with topical treatments. So transient, very mild symptomatology. And we didn't have any patients that developed any of the, I guess, the spectrum of toxicities that really have become synonymous with engineered therapies such as CAR T-cell based therapies. For example, we had no patients that developed cytokine release syndrome, no neurotoxicities. And so in summary, this product was extremely well tolerated in our patient population and indeed was associated with significant benefit. Here I'm summarizing the overall clinical outcomes in our patient population, where we achieved a 93% response rate across the board. And we achieved responses in patients who had infections with all of our target viruses, and including, as shown here, if I can show you to the second bar on the left-hand side of this graph, in 11 patients who actually had infections with 2 or more of our target viruses. And I think this really showcases the power of our therapy very carefully. We have built here a multivirus specific T-cell therapy to deal with the multiplicity of viruses that are most clinically problematic in this patient population and which can actually occur all at the same time, or actually these patients can have multiple sequential infections with these viruses, and really showcases the need for a multi-virus specific therapy, and also I think illustrates the power of our therapy with dealing with these multiple viruses. I want to speak specifically to the power of this therapy when dealing with diseases. And again, from the beginning of my slides, you'll note that I think this is really where the greatest clinical need is in this patient population. And again, as noted in this proof of concept study, primarily we were dealing with patients who had quite advanced infections, many of whom had developed diseases associated with each of these target viruses. And here I want to highlight 20 patients who had actually developed hemorrhagic cystitis that was associated with persistent BK infections who were part of our clinical trial. In this patient population, upon administration of our viral MM product, you'll see even within 2 weeks of a single infusion of our viral MM product, 60% of our patients had entirely resolved their disease. And of course, the numbers just improved from there on out. And here I'm showing you a time course over the 6 weeks post-infusion of our viral MM products. And I suppose really, the question is, how clinically meaningful is this to patients? And I want to showcase you, I suppose, a contrasting patient population. So these were patients who were treated at one of the same hospitals where we were administering patients with our viral MM product. But in this particular patient population, this was a patient population where they just received standard of care measures. So they did not receive our viral MM product, just received the supportive measures that I outlined previously. And in this particular patient population who had similarly balanced disease characteristics as our viral MM cohort, you see at the week 2 time point with just standard of care approaches, essentially just 6% of patients had resolved their disease, and even 6 weeks of standard of care measures, less than 40% have resolved their disease. So dramatically showcasing the clinical benefit associated with our therapy versus conventional approaches. And so with that, I want to just briefly introduce you to one of the studies that the company will be advancing to the clinic to truly, I think, try to exploit the full benefit of our therapeutic. And this is our viral MM registration study for virus-associated hemorrhagic cystitis, which will be initiated later this year. This is going to be a phase 3 multi-center double-blind placebo-controlled study to assess the safety and efficacy of viral MM compared to placebo controls for the treatment of patients with virus-associated hemorrhagic cystitis. Again, our patient population will be those with relatively severe disease, at least grade 3 or higher hemorrhagic cystitis. These patients will have viruria and dysuria, will be with pain, and will have associated symptoms that are associated with hemorrhagic cystitis. And in this randomization, which is balanced at 3:2, patients will receive either a fixed dose of our viral MM product or placebo. We will be administering patients with 2 infusions of our cells to really maximize the potential for therapeutic benefit. And we will be, of course, looking at safety and efficacy and following these patients for at least 6 months. So with that, I've briefly introduced you for our first clinical trial of viral MM in virus-associated hemorrhagic cystitis, but of course we have plans to exploit this therapeutic for a spectrum of the viruses that can be targeted with our multi-virus product, including a clinical trial which we will be advancing to the clinic for the treatment of CMV, for adenovirus. We will also be moving forward with a prevention study. So this will be the use of our multi-virus product to prevent infections associated with one or more of our target viruses in our allogeneic stem cell transplant population. And we will be advancing our viral MM product for the treatment of infections associated with BK and CMV in the solid organ transplant setting. But we're also moving forward with a number of our other programs, including our ALVR 106 multi-virus product targeting RSV, influenza, parainfluenza, and human metapneumovirus. These are all respiratory viruses that significantly impact not only the stem cell and solid organ transplant population, but also high-risk vulnerable populations, including the elderly and the very young. And we have plans to move that forward to the clinic in the very near future. Also, we have developed a therapeutic for the treatment of COVID-19, which we're moving forward with plans to test again in the high-risk patient population with a number of high-risk features that render them susceptible to the worst outcomes associated with COVID-19. And then 2 earlier stage products that we are developing for HBV and HHV8, ALVR 107 and 108 respectively. And so with that, I am glad to have had the opportunity to introduce you to our company and our off-the-shelf virus-specific T cells, and I thank you for your attention.